Plant Growth and Development, Part 2. Plant growth regulators. Characteristics. The small simple molecules of diverse chemical composition are plant growth regulators. They could be indole compounds, adenine derivatives, derivatives of carotenoids, terpenes, or gases. In literature, plant growth regulators are variously described as phytohormones, plant hormones, or plant growth substances. In a living plant body, based on their functions, the PGRs can be broadly divided into two groups. In growth promoting activities, one group of PGRs are involved, such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation, and these are also called plant growth promoters, example auxins, gibberellins, and cytokinins. In plant responses to wounds and stresses of biotic and abiotic origin, the other group of PGRs play an important role. In various growth inhibiting activities such as dormancy and abscission, they are also involved, and the PGR abscisic acid belongs to this group. The gaseous PGR, ethylene, which is largely an inhibitor of growth activities, could fit in either of the groups. The discovery of plant growth regulators. The discovery of each of the five major groups of PGRs, interestingly, have been accidental. All this started when Charles Darwin and his son Francis Darwin observed that in canary grass the coleoptiles responded to unilateral illumination by growing towards the light source. The site of transmittable influence was the tip of coleoptile that caused the bending of the entire coleoptile, and this was concluded after a series of experiments. In oat seedlings from the tips of coleoptiles, F.W. Wendt isolated auxin. A fungal pathogen, Gibberella fujikuroi, caused the bakani, a disease of rice seedlings. The appearance of symptoms of the disease was reported by E. Kurosawa when the uninfected rice seedlings were treated with sterile filtrates of the fungus. Later, gibberellic acid was identified as the active substance. From the internodal segments of tobacco stems, F. Skoog and his co-workers observed that in addition to auxins, the callus, which is a mass of undifferentiated cells, proliferated only if the nutrient medium was supplemented with one of the following: DNA, coconut milk, yeast extract, or extracts of vascular tissues. Later, Skoog and Miller identified and crystallized the cytokinesis promoting active substance that they termed kinetin. Three independent researchers during mid-1960s reported chemical characterization and purification of three different kinds of inhibitors: inhibitor B, abscission 2, and dormin. All the three later were proved to be chemically identical, and it was named abscisic acid. Cousins confirmed that the ripening of stored unripened bananas was hastened by the release of a volatile substance from ripened oranges. Later, this volatile substance, a gaseous PGR, was identified as ethylene. In the next session, let us study some of the physiological effects of these five categories of PGRs. Physiological effects of plant growth regulators. Auxins. Auxins from human urine was isolated first. The term auxin, having certain growth regulating properties, is applied to the indole 3-acetic acid and to other natural and synthetic compounds. Auxins are produced generally by the growing apices present in the stems and roots, from where they migrate to the regions of their action. From plants, Auxins have been isolated like IAA and indole butyric acid. NAA and 2,4-D are synthetic auxins. In agricultural and horticultural practices, all these auxins have been used extensively. In stem cuttings, they help to initiate rooting, an application widely used for plant propagation. For example, in pineapples, auxins promote flowering. At early stages, they help to prevent fruit and leaf drop, but in older mature leaves and fruits, they promote abscission. The growing apical bud in most higher plants inhibits the growth of the lateral buds, a phenomenon called apical dominance. Usually, the removal of the shoot tips results in the growth of lateral buds. In tea plantations, hedge making, it is applied widely. Can you explain why? For example, in tomatoes, auxins also induce parthenocarpy. As herbicides, auxins are widely used, and to kill dicotyledonous weeds, 2,4-D is used and it does not affect mature monocotyledonous plants. By gardeners, it is used to prepare weed-free lawns. Auxins help in cell division and also controls xylem differentiation. Gibberellins. Another kind of promotory PGRs are the gibberellins. From widely different organisms, example fungi and higher plants, more than 100 gibberellins have been reported. They are denoted as GA1, GA2, GA3 and so on. However, one of the first gibberellins to be discovered was gibberellic acid and remains the most intensively studied form. All GAs are acidic. In the plants, a wide range of physiological responses was produced by them. They have the ability to cause an increase in the length of axis, and this is used to increase the length of the stalks of grapes. In fruits like apple, to elongate and improve its shape, gibberellins are used. They also delay senescence, due to which the fruits can be left on the tree longer so as to extend the market period. In brewing industry, to speed up the malting process, GA3 is used. In their stems, sugarcane stores carbohydrates as sugar. Length of the stem increases by spraying sugarcane crops with gibberellins, thus increasing the yield as much as 20 tons per acre. Spraying GAs on juvenile conifers hastens the maturity period, thus leading to early seed production. In beet, cabbages, and many plants with rosette habit, gibberellins also promotes bolting. Cytokinins. On cytokinesis, cytokinins have specific effect, and from the autoclaved herring sperm DNA they were discovered as kinetin. Naturally, kinetin does not occur in plants. With cytokinin-like activities, search for natural substances led to the isolation of zeatin from coconut milk and corn kernels. Several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified since the discovery of zeatin. In regions where rapid cell division occurs, natural cytokinins are synthesized. For example, root apices developing shoot buds, young fruits, etc. It helps in the production of new leaves, chloroplasts in leaves, lateral shoot growth, and adventitious shoot formation. Apical dominance is overcome by the help of cytokinins. It helps in the delay of leaf senescence as they promote nutrient mobilization. Ethylene. Ethylene is a simple gaseous PGR. In tissues undergoing senescence and ripening of fruits, it is synthesized in large amounts. On plant influences of ethylene include apical hook formation in dicot seedlings, horizontal growth of seedlings, and swelling of the axis. On plant organs, especially in leaves and flowers, ethylene promotes senescence and abscission. In fruit ripening, ethylene is highly effective. During ripening of fruits, it enhances the rate of respiration. The rise in the rate of respiration is called respiratory climactic. Breaking of seed and bud dormancy, initiation of germination in peanut seeds, and sprouting of potato tubers are done by ethylene. In deep water rice plants, ethylene promotes rapid internode petiole elongation, which helps leaves or upper parts of the shoot to remain above water. Root growth and root hair formation is promoted by ethylene, thus increasing the absorption surface. To initiate flowering and for synchronizing fruit set in pineapples, ethylene is used. In mango, it also induces flowering. Since one of the most widely used PGR in agriculture is ethylene, as it regulates so many physiological processes. Ethiphon as a source of ethylene is the most widely used compound. In an aqueous solution, ethiphon is readily absorbed and transported within the plant and slowly releases ethylene. In tomatoes and apples, ethiphon hastens fruit ripening, and in flowers and fruits it accelerates abscission. In cucumbers, it promotes female flowers, thereby increasing the yield. Abscisic acid. Abscisic acid, as mentioned earlier, was discovered for its role in regulating abscission and dormancy, but it also has other wide-ranging effects like other PGRs on plant growth and development. It acts as an inhibitor of plant metabolism and a general plant growth inhibitor. ABA inhibits seed germination. ABA increases the tolerance of plants to various kinds of stresses and also stimulates the closure of stomata in the epidermis, hence it is also called as a stress hormone. ABA in seed development, maturation and dormancy plays an important role. ABA helps seeds to withstand desiccation and other factors unfavorable for growth by inducing dormancy. ABA acts as an antagonist to GAs in most situations. For any and every phase of growth, we may summarize that for differentiation and development of plants, one or the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, in the life of a plant, there are a number of events where to affect that event more than one PGR interact. Example, dormancy in seeds or buds, senescence, apical dominance, abscission, etc. Remember, only one kind of intrinsic control is the role of PGR. They play an important role in plant growth and development along with genomic control and extrinsic factors. Temperature and light, which are some of the extrinsic factors that control plant growth and development via PGR. Vernalization, flowering, dormancy, seed germination, plant movements, etc. could be some of such events. Photoperiodism. It has been observed that to induce flowering, some plants require a periodic exposure to light. It is also seen that the duration of exposure to light is measured by such plants. For example, the exposure to light for a period exceeding a well-defined critical duration is required by some plants, while for a period less than this critical duration, other plants must be exposed to light before the flowering is initiated in them. The later group of plants are termed short day plants, while the former group of plants are called long day plants. The critical duration for different plants is different. However, where there is no such correlation between exposure to light duration and indication of flowering response, there are many plants. Such plants are called day neutral plants. Now, it is also known that the duration of light period and the duration of dark period also is of equal importance. Hence, it can be said that in certain plants, flowering depends on a combination of light and dark exposures and also on their relative durations. To periods of day or night, this response of plants is termed photoperiodism. It is also interesting to note that prior to flowering, shoot apices modify themselves into flowering apices, while by themselves shoot apices of plants cannot perceive photoperiods. The leaves are the site of perception of light or dark duration. It has been hypothesized that for flowering there is a hormonal substance that is responsible. This hormonal substance for inducing flowering migrates from leaves to shoot apices only when the plants are exposed to the necessary inductive photoperiod, vernalization. On exposure to low temperature, there are plants for which flowering is either quantitatively or qualitatively dependent, and this phenomenon is termed vernalization. Late in the growing season, it prevents precocious reproductive development, and to reach maturity, it enables the plant to have sufficient time. Especially, vernalization refers to the promotion of flowering by a period of low temperature. Some important food plants like wheat, barley, rye have two kinds of varieties, namely winter and spring varieties. Normally, in spring, the spring variety are planted and before the end of the growing season, they come to flower and produce grain. However, winter varieties would normally fail to flower or produce mature grain within a span of a flowering season if planted in spring, and hence they are planted in autumn. They germinate and come out as small seedlings over winter, resume growth in the spring, and usually around midsummer they are harvested. In biennial plants, another example of vernalization is seen. Biennial are monocarpic plants that normally in the second season flower and die. Some of the common biennials are sugar beet, cabbages, carrots, etc. To a cold treatment, subjecting the growing of a biennial plant stimulates a subsequent photoperiodic flowering response. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.